Hello everyone, welcome back to another video tutorial series. I hope you all are in good health. Before starting this session, I can share my perspective about the global crisis that we all are a part of it. Don't panic but be prepared. Follow command from higher authority. Keep social distance. Protect our vulnerable. Self-isolation as necessary. Finally, stay healthy. Let's start our session. This will be a series of video on how nanoparticles used for virus detection. If you want to gain some knowledge about what is virus, its different types, current and past detection options, and finally how nanotechnology is used for virus detection. What is virus? Viruses are remarkable pathogens that are causing prominently increasing morbidity and mortality worldwide. Their highly contagious nature and the absence of immediate and efficient control systems are the main reasons behind their potential health impacts. Currently, viral infections and associated diseases are major causes of death in mankind. Viruses are particulate in nature and usually exist in different morphological forms that generally range in size from 20 to 900 nanometers. Their mature infectious particles are typically composed of definite units of proteins and nucleic acid that self-assemble to form nanoparticulate structures called virions. The viral proteins are usually arranged in a surface layer called the capsid and sometimes in an outer envelope surrounding an inner core of nucleic acids. This core of viral nucleic acids can be single or double-stranded, DNA or RNA, one or several molecules, linear or circular in shape, and a few thousands of nucleotides to 1 million base pairs in size. Classification of viruses. Viruses are grouped on the basis of size and shape, chemical composition and structure of the genome, and mode of replication. In the past, many outbreaks of viruses, such as human immunodeficiency virus, HIV, severe acute respiratory syndrome, SARS, influenza virus, H5N1 and H1N1, and Zika virus. Currently COVID-19. Past and current virus detection options. The first studies on virus isolation and detection were started early in the 1950s when the first cell culture system and electron microscope were developed. Serology remains the standard method for virus detection. It primarily relies on testing for the presence of specific viral antigens or the corresponding antibody responses of the immune system. The most common types of serological tests include the neutralization assay, complement fixation test, immunoprecipitation assay, IPA, hemagglutination inhibition, high assay, enzyme immunoassay, radioimmunoassay, RIA, chemiluminescent immunoassay, CIA, particle agglutination, immunostaining, immunofluorescence assay, single radial hemolysis, immunoblotting assay, IBAR, and immunochromatographic test, ICT. Molecular techniques are attracting more interest and have found an increasing number of applications in virus detection. These techniques can be classified as amplification or non-amplification based molecular detection techniques. Amplification-based molecular techniques usually employ one or more forms of nucleic acid amplification to allow the indirect detection of the target virus. These techniques represent the majority of molecular techniques applied in virus detection and include various types of target amplification techniques. For example, PCR, loop-mediated isothermal amplification, LAMP, transcription-mediated amplification, and nucleic acid sequence-based amplification, signal amplification techniques. For example, branched DNA and hybrid capture, and probe amplification techniques. For example, ligase chain reaction and strand displacement amplification. Non-amplification-based techniques are mainly applied to the direct testing for the presence of a specific virus in clinical samples, for example, in situ hybridization, southern blot hybridization, and blot hybridization. Nanotechnology patent virus detection. Nanomaterials are known to possess multiple unique optical, electronic, magnetic, and mechanical properties enabling very attractive applications, especially in the fields of biomedical imaging and clinical diagnosis. The first reported application of nanomaterials in the detection of viruses was attempted in the late 1990s. AMPs were coupled with silver staining and applied for the detection of human papillomavirus in cervical carcinoma cells. Currently, there is a very wide range of nanomaterials, including metal NPs, carbon nanotubes, silica NPs, quantum dots, QDs, ARP conversion NPs, and polymeric NPs, that are being heavily investigated for virus detection. Thank you for watching this video. I hope you will gain some basics. If you like this video tutorial, share with your friends. Sharing your knowledge with others does not make you less important. I wish you all good health.